Hey everybody, we are continuing to look at the types of living things that are out there in our overall study of biology. We've done a lot of look at some very small things with cells, genetics. We've looked at how things like traits are passed down from one generation to the next. We've talked about how that can affect individuals as well as populations and even species. And we've kind of mentioned along the way how these individual species are named specific things, and they're named binomially. They have those two-part names, a genus and a species, and that's really what we're going to push into next. Right, so we're looking at the kingdoms of life. For our purposes, we're going to stick with one of the systems. There are a few systems out there, but let's go ahead and get into it. For 7.1, we're thinking about this organizational system, but first we're going to talk about something called cladograms and dichotomous keys. Alright, so cladograms and dichotomous keys both fit into the study of phylogeny and cladistics, which are related studies. Right? They both focus on trying to categorize living things into groups, even way back in Darwin's studies on the beagle. Right? We've got this sketch here. It's actually from one of Darwin's notebooks that he had with him at the Galapagos Islands. Right? And you see this branching relatedness shown here. We might think of it kind of like a idea map or a concept map, right? Or like a family tree, right? They are just a visual representation, and now in modern science we use these to talk about evolutionary processes and progress, and they are usually based on DNA similarities now, but they're also based on physical structures. And that's how we're going to use them in my class, is physical structures, right? But one of the scientists that really kicked this off, he was a naturalist. Remember, that was our word for biologist before biologist was a word, right? His name was Ernst Haeckel. He was a German adventurer, really. He went out into the wilds and captured animals to study them, because that's how you studied animals in the 1800s, right? He took Darwin's work in the late 1800s and really popularized it in Germany. He spoke about it a lot. He published more works supporting it and backing it up. He did a lot of these graphic representations, like the one you see on the right, in his books, right? And this visual organization helped a lot of people understand the relatedness, because they were used to family trees. They were used to lineages when talking about like royal families or just your own family. Right? Moving a little bit closer to the modern day, we have Emil Hans Willi Hennig. Right? He goes by Willi Hennig for most of his adult life. The Emil Hans part gets dropped. You do see, now we're using the term biologist, right? We're in World War II. He's working during and after World War II, right? A lot of scientists were working for Germany during World War II. A lot of them moved to NASA afterwards. Emil stays in Germany. He is part of Western Germany, right? And he focuses this study on genealogies and how they can be showing the relatedness as evolution occurs, right? But it's also using these new informations on DNA, right? And how we can start to look at how DNA might be related, right? That doesn't get really picked up. It was an idea that he had more than anything else, but that idea gets picked up again in the 90s, right? But something that's still around today is the Willi Hennig Society. You don't have to know this, but I wrote it down so that I would remember it when I was talking about it. It is a society much like the Linnaean Society that we've talked about with Darwin and Wallace. Right? They are more focused on cladistics, though. Right? So, some things to remember. A phylogenetic tree, a cladogram, are pretty much the same thing. Right? And we're going to use those terms interchangeably, but they're not actually the same. Phylogenetic trees are really evolutionary hypotheses, whereas a cladogram is on a species' similarities, right? So we're going to be mainly studying cladograms, but you might hear them called phylogenetic trees sometimes. It's okay to use those interchangeably for all intents and purposes. However, I do want you to know that they are technically different. Right? Both are trying to show the change in an organism over time. They just approach it from different methods. Right? So remember, phylogenetic trees, evolutionary hypotheses. Cladograms, just looking at a species' similarities. Right? So what do we mean when we say species similarities? Right? So these are going to look at traits that they have in common. Right? So you see this example on the right side. Right? It's got insects, fish, amphibians, birds, prosimians, which is like the lemur here, and humans on the far right side. The blue text here is showing traits that they have in common. So the farther up you go and to the right, in this case, the more of those traits they have. So all of these develop from a blastula. That's the small clump of cells after sperm meets egg in humans, right? But insects don't have vertebrae. Everything else does. Fish are not tetrapods, meaning they have 4 limbs, but amphibians, birds, prosimians, and humans do. Right? The amniotic egg isn't true for amphibians, fish, and insects, but is true for birds, prosimians, and humans. Of course, prosimians and humans are the only ones with hair, and humans are the only ones that are bipedal. So we're seeing these traits being shown. Right? And if we were to follow these down, we could see where they fork and split. That's where we would call something called a more recent common ancestor or a less recent common ancestor. Right? That common ancestry is the point at which, evolutionary speaking, secular scientists are saying they're going to split. All right? Remember, we're just looking at secular scientists and saying this is a thing that they believe. That way we can talk about it and converse about it and know what they're talking about so that we don't sound silly or uneducated. Right? Now, if we take that information, we can also split it to look more like a tournament bracket. Right? And I know that you can't see this very well. I'm just kind of showing it to you for text. We're going to go over a lot of these in class. Right? But you can see here we've got a couple of things added to this. So this tree has organisms A, B, C, D, and E. Right? If we move farther back where there's less concurrent species and there are more ancestor species, those are going to be ancestors. While we go towards the more branched side, those are the present day ones. Right? The very, very, very first one is going to be called a root. Right? And it's going to have the most recent ancestor of all of them. Right? So the more we split it up, the less common ancestors we're going to have, but they're going to be more recent. Right? We can also spin these around. We can change their shapes. They can be represented a few different ways. Right? So all of these trees are the exact same relationships. They're just spun. Right? We're just rotating around the branches. Now, this does make Mr. Baker's grading job a little harder, but it's okay. For the most part, I have all the keys possible for this kind of thing, or I can make sense of whatever you draw, right? As long as we are going with the same representation. Now, what does make it easy is that a lot of times we're going to work off of these trait tables, all right? And they're going to be organized much like this. We'll see a few of them in class, all right, and a few of them in the readings, hopefully, that you've already seen. So these are going to say the exact same thing that the cladogram or the phylogenetic tree might say, but it's represented as a table. So if we're going to draw this, it's going to look something like this. Right? Pause the video, make sure you understand it. We're going to go over these in class again, just trying to show you that all the ones in the purple-ish outer oval rectangle are vertebrates, but the ones that are in the blue square in the center, those are the only ones that have hair and fur, and if you look down at the table, you can see those same things happening. Right, so one of the ways that we can organize this is look at our trait tables and we count how many characteristics each one has. This one's color-coded, and 0 is just no, and 1 is yes. So a lot of times we'll see check marks. This one just has ones. I stole it off the internet. It's okay. All right. So use your imagination. Turn all those ones to like check boxes, and you're good to go. So you can see that the leopard only has a single trait that it has, so we know it's going to be on the far right. The lancelet has none of them, right? So it's going to be what's called an outgroup or an outlier, right? You see it splits off before the very first trait is even mentioned, right? It's not related to these very closely at all, is what a cladogram will say, a phylogenetic tree will say, right? Whereas then we start to see, okay, the lamprey has 1, the tuna has 2, the salamander has 3, the turtle has 4, the leopard has all of them, all right? So we've got this stair-step pattern if we organize them from the most traits to the least traits, right? And then we just connect the dots and add the traits labeled like that, right? We're going to go over a few of these in class, so you can pause the video and try these yourself if you would like now, and we'll talk about them in class. But that one turns out to look like this, right? Dichotomous keys, the next big thing, right? It is effectively a bunch of yes or no questions that allow us to identify a species. We're going to do one with fish in class, but it's just kind of like a choose your own adventure book. So you go to the first question. It says, has a green colored body or has purple colored body, and depending on what color it is, you're going to go to a different question. So you follow that question all the way down until you get to the end, or you get to one that identifies it. Right? So these can be kind of complicated, they can be kind of short. It depends on what you're trying to identify. So we've got a couple of different new science tools. We're going to use them in class, right? But for now, know that we try to organize them based on their traits, and secular science is also going to try to organize them based on evolutionary relationships as hypothesized, right? And I will see you guys in class.